CRISPR Gene Editing. Certainly, CRISPR, clustered regularly interspaced short palindromic repeats, is a revolutionary gene editing technology that has garnered immense attention in the field of biotechnology and genetics. It allows scientists to precisely modify the DNA of living organisms, including plants, animals, and even humans. The CRISPR system was originally discovered as a natural defense mechanism in certain bacteria against viral infections. It consists of two main components, the Cas9 protein and a guide RNA, gRNA. The gRNA is designed to target a specific sequence of DNA that needs to be edited, and the Cas9 protein acts as a pair of molecular scissors to cut the DNA at that location. Once the DNA is cut, the cell's own repair mechanisms come into play. Scientists can either disable or replace specific genes or insert new genes at the target site. This ability to make precise changes to the genetic code has vast potential for various applications in medicine, agriculture, and biotechnology. CRISPR gene editing holds promise for treating genetic disorders by correcting or removing faulty genes responsible for diseases. It also offers the possibility of creating genetically modified crops that are more resistant to pests, diseases, and environmental stresses, potentially leading to improved agricultural productivity and sustainability. However, CRISPR technology also raises ethical concerns and regulatory challenges. The ability to manipulate the human genome raises questions about the potential for misuse, unintended consequences, and the creation of designer babies. CRISPR research was ongoing, and scientists and policymakers were carefully navigating the complexities of its applications. Since then, there might have been significant advancements or updates, so it's always best to consult the latest sources for the most current information on CRISPR gene editing. Don't forget to like, share, and subscribe for more exciting content on the latest technology trends and innovations. Thanks for watching.